So it's my great pleasure to introduce Karen Allen, this year's Green Lecture in Enzyme Chemistry from Boston University. First, a little bit about David Green. And so this is a lecture that lectureship that was established in his honor. So David Green came to UW-Madison from Columbia to co-direct and helped found the Enzyme Research Institute, which is closed now, but for many of you know is just down the street next to one of the Wisconsin Foundation buildings, and it still says Enzyme Research Institute on the outside. He was an extremely prolific biochemist, particularly in the areas of metabolism. He's very well known, as it says up here, for working on pyruvate oxidation and fatty acid oxidation. A lot of the, if you read about this, a lot of the tissues were donated by Oscar Mayer, which had a plant locally here in town near the airport up until just about maybe 8 or 9 years ago now. During his time at Madison, his group published 559 scientific publications, which is pretty amazing over 33 years. Science and politics also ran in his family. So his daughter became a very well-known enzymologist by the name of Rowena Matthews, who just retired a few years ago. But she was at University of Michigan for a long time and worked on vitamin B12 utilizing enzymes. And he raised his granddaughter, who is Tammy Baldwin, who is the Wisconsin senator and who's one of the people kind of leading the fight to maintain science funding in Washington and kind of working against the leadership there. So this is really fitting that Karen is here to give today's lecture. So her kind of pedigree in enzymology is outstanding. And so she received her PhD from Brandeis, working with Bob Abels, who, as many of you know, was really one of the pioneers of mechanistic enzymology in the mid 20th century. My PhD advisor also was in the Abels lab, actually. Then after that, she switched gears a bit to work on structural biology with Greg Petsko. And Greg Petsko is also one of the pioneers of structural biochemistry, particularly of enzymes and studying their structure-function relationship. After that, she established her own lab at BU. I was mentioning to Karen earlier, the first time we met was many years ago, and she didn't remember this, but this is etched in my memory. So the very first conference I went to as a graduate student was the Enzyme Mechanisms Conference in Galveston, Texas. And I presented a poster there, and so I put up my poster, and you know, they have talks and stuff, and we go to the poster session, and I look down the hallway, and like everyone who's anyone in enzymology was like standing like at my poster. It was like Tom Bruce, Stephen Bankovic, Chris Walsh, Perry Fry, I'm sure was there, Mo Cleland. And I get closer and I realize they're actually all wanting to see Karen's poster, which was right next to mine. So she had this really interesting structure of phosphoglucomutase where there was this interesting intermediate crystallized with a highly unusual and very thought-provoking three-dimensional structure. Since then, she's really determined the structures and elucidated mechanisms of many enzymes, many of which are fundamental to biochemistry and metabolism. For those of you who take 507, you will hear about some of these. And so I already mentioned phosphoglucomutase. One of the structures of Karen's that I usually went over in 507 was acetoacetate decarboxylase, which is a paradigm for thinking about how enzymes can use perturbed pKa's to catalyze biochemical reactions. And with that, I'll turn things over to Karen and welcome her to Wisconsin.
Thank you so much. It's truly an honor to be here to give the Green Lecture. And, you know, it's a wonderful legacy, and I know Rowena Matthews, and it's just a fantastic thing to be invited to do. I really have enjoyed my day here today, the faculty that I've met and the really vibrant students here. It's really fantastic. So I want to tell you about this work that my lab has done together in collaboration with the lab of Barbara Imperiali at MIT on the enzymes that do make the giant glycans that decorate the outside of bacteria. And so what I hope I'm going to get you to think a little bit about is thinking a little beyond the active site and what are the determinants that can address specificity in enzymes. And I want to start out my talk just thanking those who did the work, because this always seems like an afterthought later, but really the lab, my lab, the students have been fantastic and very hardworking. The Imperiali lab at MIT, we've done all of this work very much in the way of a sort of Vulcan mind meld, bringing our labs together. And so I also, of course, want to thank Brookhaven, Argonne, and SSRL for photons, and the NIH for funding this work. So bacterial glycoconjugates occur across species in both gram-negative and gram-positive bacteria. And really these glycoconjugates decorate the outside of bacteria. So you see in the gram-negative bacteria, lipopolysaccharides, capsular polysaccharides, the N-linked glycoproteins, which will be the focus of our attention today, and also actually decorating the flagella. And then in the gram-positive bacteria, making the teichoic acids like the lipoteichoic acids and wall teichoic acids. And really, when we think about these, I want to focus your attention on the N-linked glycoproteins. These are very important because when they do decorate a protein, what's happening there is really these are going to add additional specificity elements and also additional elements that change the physicochemical properties of the protein itself. And so the glycan that we'll focus on today is the biosynthesis of the glycans in Campylobacter. And specifically, the one that I've drawn here is the N-linked heptasaccharide that is in Campylobacter jejuni. And in this heptasaccharide, what we can see is that we've got both 1,4 linkages, and then this sort of little side chain 1,3 linkage, making up that saccharide that will eventually be decorating these types of proteins in bacteria. Okay, so. Oh, wait, I can use my slide advancer. Okay, so let's just go over the pathway by which we make these oligosaccharides. So it's an en bloc synthesis with an initiation step, an elongation step, and finally a transfer step onto the final destination. And so what we see is that in Campylobacter jejuni and Campylobacter concisus, these kinds of bacteria are really the common cause of human intestinal tract diseases. There's this same pathway occurs in H. pylori and also in gonorrhea. These are bacteria you really do not want living in and on you. And so possibly doing therapeutic intervention into this pathway would be a good way to design antibacterial agents. What we see is that the kinds of glycan that I drew for you earlier is actually found on 65 different proteins in Campylobacter. And if you shave off those glycans or impair their glycosylation, you find decreased pathogenicity. Okay, so let's go over then the pathway. So we always start out with an initiating enzyme, and this is a very important first step, because what this first step does is it dedicates the glycan growing to the membrane. And so the very first step is to put a sugar phosphate group onto an undecaprenol phosphate carrier. Now, the same type of pathway does happen in humans, but our carrier is dolichol, whereas in bacteria, it's always these undecaprenols. Every enzyme in the pathway is also membrane embedded. And so this entire pathway is happening like an assembly line on the membrane. The first step then is to take a nucleoside substrate, nucleoside sugar substrate, and to attach a phospho sugar onto the undecaprenol phosphate, kicking out UMP. After that first initiation step, we have 3 elongation steps that are carried out by GTB folds or glycosyltransferase type B folds. Those folds then will kick out instead a UDP. So they're not doing a phospho-sugar transfer. They're just transferring a sugar. Okay, so we get sugar, sugar, sugar. The third enzyme in the jejuni pathway actually does a precessive reaction where it's putting on 3 glycans in a row. Okay, so we have 2 single steps and a precessive step. Now a GTA type fold will put on that little side chain. And it's really interesting that nature has changed the type of fold to do that slightly different stereochemical step. Then a floppase will take this entire glycan chain and flip it to the other side of the membrane, where it will eventually be moved to the protein target by the enzyme PGLB. And so what you can see is this entire assembly line is started on the membrane by this very first enzyme, PGLC. So that enzyme is the important initiating enzyme. Now, looking at the glycans across all bacteria that are made by this same pathway, we can see that there's a huge amount of diversity. I'm not expecting you to actually look at this slide. It is called a shock and awe slide. You're supposed to be awed by the sheer diversity of the bacterial glycans that are made, even in very similar types of bacteria. So we have this great diversity of glycans. All the ones where I've shown as white squares are actually have an unknown sugar in that place. And so you can see that there's a great deal that has yet to be found out about these types of pathways. And our big picture question is really, can we find structure function signatures so that instead of analyzing these by growing bacteria, shaving the glycans off and doing mass spec, which is very difficult to do because many of these sugars are actually isobaric. They have the same mass and we can't tell them apart, hence all the white squares. And so what we would really like to do is to be able to take the enzyme sequence and read from it the signature and read the sequence determinants, be able to know then what the final glycan product will be. And so to address this, we really focused on the Campylobacter. And the reason is because Campylobacter has done us a big favor. They've organized their biosynthetic pathway for these end-linked glycans into operons. So all the genes that do the pathway are all located together. And not only are they together, they're in the same order. And in a very diverse superfamily of proteins, ortholog identification can be very difficult because the sequence identity is quite low. But because they're in the same order, when we're comparing 2 bacteria to one another, we always know which enzyme is the PGLA or the PGLC or the PGLH. And so we know the operons, we can pull them out with the signature phosphoglycosyltransferase, and then be able to identify those enzymes within the pathway and put them in order. Okay, so with that as our introduction to the beast that we will be studying, what we want to start out with is that important initiating enzyme, PGLC in Campylobacter jejuni and Campylobacter concisus. So this is just a little preview of what the structure looks like. And we wanted to really focus in on this as being a target for our structural and functional work to begin with. And I just want to point out that these Campylobacter always use an unusual sugar to do that initiating pathway reaction. And that is N-N-acetyl-bacillosамine, which I will lovingly call NN-DINAC-BAC for the rest of the talk. Okay. So just to remind you of what the chemistry is here, what we're going to do is we're going to take our sugar nucleoside, we're going to then do a transfer onto this UMP, onto this undecaprenol phosphate. We're going to kick out UMP and get our final product, which is membrane embedded. So we have a couple of features here. One is that one of our substrates is very soluble. The other one is completely membrane embedded. Okay, and so we need a way to bring these two things together in the same active site. And again, the uniqueness of this reaction can also allow us to identify this pathway in operons. Now, nature has played us a rather nasty trick, and that is that there are actually two enzyme fold families that can do the same phosphoglycosyl transferase reaction. Okay, there is the polytopic PGTs, which have 10 to 11 transmembrane helices. They're best typified by MRAY and WEC-A. They're found in both prokaryotes and eukaryotes. So these are the types of enzymes that do that first reaction in humans, okay? For these, nucleoside analogs like tunicamycin are actually potent and good inhibitors. However, there are also the monotopic PGTs. They do the same reaction I showed you on the previous slide. These have a single membrane embedded helix. They're typified by our PGLC. And for these, tunicamycin is a very poor inhibitor. So another question we're going to try to answer in our talk today is just why would nature use two scaffolds for the self-same reaction? Okay. Now, looking further into these two scaffolds, we see that not only are they structurally divergent, they do the same reaction, but they're mechanistically divergent. So for the polytopic PGT, we see that it goes through a ternary complex mechanism. It will bring the polyprenyl phosphate and the UDP sugar together into the active site at the same time for a direct attack of the polyprenol onto the phosphoryl group of the nucleoside sugar, kicking out UMP and giving us our final product. However, in work by the Imperiali lab, we see that for the monotopic PGTs, it's a very different mechanism. Instead, it goes through this phosphoglycosyl intermediate. Okay, so we have a nucleophilic aspartate that attacks the phosphoryl group, giving us this phosphoglycosyl intermediate and kicking out UMP in the first half reaction. In the second half reaction, we have the attack of the polyprenol, our undecaprenol phosphate, from the membrane into this phosphoryl group, liberating our enzyme nucleophile and giving us our final product. Now, this was shown by the Imperiali lab in classic type of enzymology experiments where you can do a sodium borohydride reduction and show that the aspartate is modified to be a homoserine. You can also take the enzyme and run it backwards in the presence of labeled UMP and regenerate a labeled substrate, showing the intermediacy of this of the phosphoglycosyl intermediate. And of course, Mo Cleland's famous ping pong kinetics. So two half reactions shown here, very different kinetics. Well, these kinetics also explain the difference between the nucleoside antibiotics such as tunicamycin acting on these two different scaffolds. Remember that I said for the polyprenol, it's a ternary complex mechanism. So this type of inhibitor like tunicamycin with a Pren-P analog, a sugar analog, and a nucleoside analog is actually a very good inhibitor. It's a bisubstrate analog. Whereas for our little PGT that has a monotopic membrane protein, in that one, what's happening is that the UMP has already left after the first half reaction. These two substrates are never in the active site at the same time. So tunicamycin is a terrible inhibitor, and that explains the difference between them. Okay, so we have two very different scaffolds. We're going to focus then on the monotopic PGTs that are poorly inhibited by tunicamycin. Those are the PGTs in the C-Jejuni pathway that I showed you earlier for the N-linked glycosylation. Okay, so the first thing that my lab did in looking at this problem was to start with some bioinformatics where we found all 63,000 members of the monotopic PGT family, and we did a giant sequence alignment. And what you can see is a couple of things. One is that there's this absolutely conserved DE motif. That's our nucleophilic aspartate that I showed you in the mechanism earlier, and an adjacent glutamate. And so that's our asp glue dyad, which is absolutely invariant throughout the family. We also have this PRP motif, which I'll show you later. It can be important in stabilizing the phosphoryl groups during the reaction. There will be a quiz, so remember that PRP motif. All right, the other thing we can see is we can divide our superfamily into 3 subfamilies. There's the small PGTs, which are really just the catalytic domain, undecorated. We also have the bifunctional PGTs, which are the catalytic domain, which is decorated with either enzymes previous in the pathway or enzymes later in the pathway. So some of these are fusion proteins. Third, we have the large PGTs, which have an additional 4 transmembrane helices, and then a Rossmanoid fold domain of unknown function, which we actually believe may be involved in regulation. We can talk about that at some point. Okay, so my lab approached this problem of getting the structure of the monotopic PGT from C. concisus. And what we did is we just used detergent solubilized enzyme, and we got these pretty crystals that I show you up here, and this rather unusual fold. So what you can see is the signature reentrant membrane helix. So it goes down and then back up again. So it's not like a lollipop going through the membrane. And then we have the cytosolic domain up here. Now, I learned to love AlphaFold because we couldn't see the last 20 amino acids in the structure. It was sort of some disordered density. But later on, looking at the AlphaFold model, we saw that actually we could fit the C terminus of the protein into that density. We just needed a little bit more guidance as to what it would look like. And so we actually redeposited the structure with the additional 20 amino acids on the end. So you can see here now the complete structure of our C. concisus enzyme. So this should be in the membrane, and so we use the PPM server to dock it into the membrane. And what you can see is that compared to sort of typical monotopic enzymes in the protein data bank, there's a very good delta G of transfer for these, this is for the monotopics in the PDB, and this is our delta G of transfer for the PGLC. So you can see that that enzyme has a good delta G of transfer. This seems like a good docking location into the membrane as shown here, okay? Again, with the reentrant membrane helix going down into the membrane, but coming back up again. We also, this is actually a novel fold. So it has this long beta hairpin associated with tightly to this alpha helix. And because it looked very much like a boat sailing through a membrane ocean, we called it the Ahab fold in that sort of nautical theme. So alpha helical associated beta hairpin fold. So we have sort of the prow of the ship, you know, where the mermaid would be, and then we have this sort of mainsail here. Okay, so does that make sense biochemically? We think that it does. If I color here with hydrophobics in red and polar residues in white, what you can see here is that the part of the membrane that has, the part of the enzyme that has the reentrant membrane helix is down here. It's very red. If I actually peel the reentrant membrane helix off and turn the bottom of the boat toward you, you can see this view, so that the helices that are sunk down into the membrane, the deck of the ship, are actually amphipathic helices, so that the bottoms are hydrophobic and the tops are hydrophilic facing the active site. And so it sort of makes sense that these helices C, D, and I are all amphipathic. The reentrant membrane helix is quite hydrophobic. So we can easily see how a substrate can float up to the membrane, which is to the active site, which is membrane embedded. But we can also see that a hydrophilic substrate can also bind to the enzyme. And that's shown here by simply doing some electrostatic coloring. And this makes sense in terms of, blue here is a very positive surface. So looking at the boat from the top, you can see that there's a very beautiful saddle for these extremely negatively charged substrates like UDP, DINAC back, and even the phosphoryl group of UNP to come and dock into the active site. Again, the nucleoside sugar coming from the solution, UNP coming from the membrane side of things. So we kind of lucked out with our first structure because what we wound up with was the magnesium, which is an absolutely necessary cofactor in this reaction. We wound up with that magnesium ion right there on our nucleophilic aspartate. We also accidentally got a phosphate group bound to the active site, but that's great because that can serve like a marker and surrogate for our phosphoglycosyl intermediate. Okay, and then here's our glue from our asp glue dyad. We could also see a PEG molecule, and remember the quiz, the arginine from the PRP motif? We can see that that too is coming into the active site. So we have this active site right at the membrane interface for this enzyme. Okay, and if you'll just let me geek out for one more second about the active site. Nature has done something really amazing. So leading from the aspartate 93, which serves as a cap for this helix in the fold, what you can see is that by it serving as the cap, we've broken the symmetry of the carboxylate group and set it up for nucleophilic attack. You can also see that actually looking down the helix, so down from this side, you can see that it's actually a 310 helix, a much more narrow helix at the beginning where the nucleophilic aspartate is, and then becomes a regular alpha helix afterwards. Well, what that does is it allows the asp glue dyad to actually be very proximal to one another because the helix is quite narrow. Whereas in a normal alpha helix, these two residues next to one another would be quite splayed apart. So it brings our asp glue dyad next to each other by having this unusual 310 helix, and it breaks the symmetry of our nucleophile, setting it up for nucleophilic attack. So now I can draw the enzyme in mechanism in its active site. And what you can see here is just my aspartate 93 attacking the phosphoryl group of our substrate. We kick out the phosphoryl group, kicking out the UMP in the first half reaction. I then bring the Pren-P in, and now once the UMP has left the building, the Pren-P is able to attack here, the phosphoglycosyl intermediate. And I've kind of drawn the glutamate in the asp glue dyad as helping out by holding on to the magnesium during that second half reaction and second nucleophilic attack. We then go ahead to make our final product. So this lets us draw our mechanism right in the active site. Now, I'm drawing this here, and you saw earlier that I did have a PEG molecule bound. Maybe that could tell us where undecaproenol phosphate is bound, but I wasn't really satisfied by this. And undecaproenol phosphate really messes up our crystals. And so we couldn't really get good crystal structure. And so what we turned to then was computational analysis. So we did molecular dynamics with our collaborators in the Straub lab at BU. And we started a structure with a structure of PGLC and undecaproenol phosphate in a membrane solution that would look very much like the bacterial membrane. We put this whole thing in a hydrated box, right? And so we started with 2 copies of undecaproenol phosphate and 1 copy of PGLC. And what this slide here is supposed to, or this segment here is supposed to show you is that for the undecaproenol phosphate, whether the PGLC is there or not, it always actually curls up and is actually occupying only a single leaflet of the membrane. And you can see that here. And so it's really interesting because it actually mirrors the topology of the protein itself. So even in the absence of it, that is the equilibrium position of our undecaproenol phosphate. And during the 500 nanosecond simulation, indeed, one UNP molecule out of the two did float up into the active site of the enzyme. And so I'm showing you that view here. And so I had left the phosphoryl group there as being the phosphate that was bound during our crystallography. I left that there as a marker for the phosphoglycosyl intermediate, which would be there in the presence of the UNP. The UNP then binds to the enzyme active site. I'm going to now turn that view toward you. And what you can see is the enzyme's almost like thrown its arm around the undecaproenol phosphate as it has entered the active site. And so this really gives us an idea of where it would be. Here's the head group of the undecaproenol phosphate. And what we did is looked then for some biochemical, chemical evidence for these molecular dynamic simulations. And so looking at the residues that are surrounding that phosphoryl head group, what we did is we found these lysines and arginines. And if we mutate these one at a time, what you find is that the enzyme loses activity, but activity is not completely oblated. So the undecaproenol phosphate can still bind, but just poorly. And so this would be expected if you remove some of the binding determinants from this enzyme. And so we think that this is what's happening here and that this is actually what's happening. Actually, if we repeat the simulation with the mutated enzymes, we find that the undecaproenol phosphate no longer docks into the active site. So there is an electrostatic component to the docking when we move these lysines and arginines that help to stabilize the binding. Okay, so that's our UNP portion. Now we also tried to get some structures with the UDP sugar bound into the active site. And we had a lot of trouble with that. We could never get really clear density. But as crystallographers, we could do something. And I'll tell you what that is. So basically, when we tried to bind the substrates into the active site, we did see density, but not good enough to build in our molecules. But what we saw instead was that the symmetry of the molecules was broken. So we actually had 8 copies of the enzyme in the asymmetric unit. And in each of the 8 copies, the loop at the prow of the ship, where the mermaid is, right? That was actually in a different conformation so that it looks like it was becoming more open or more closed. Well, we couldn't build in our molecule, but we could count electrons because you can do that if you're a crystallographer. And so what we did is we counted the total electron density, and we found that indeed, when there was a lot of electron density in the active site, we had a more closed conformation, right? Here's the distance between the loop and the active site aspartate. And then if we had less electron density, we had a more open conformation. So as enzymologists, we love this. It's a typical induced fit type of mechanism. Again, we looked at this with our collaborators in terms of molecular dynamics. And what we saw was that here's our experimental data, and that's the type of movement we saw in our experimental data. Here's the molecular dynamics simulation, and it also showed that same kind of rigid closing and induced fit type of closure. So this is just showing the computational principal components. And if I compare the experiment and the simulation in this heat map, we see that the first principal component matches up very well. So basically, we like to infer from this that there is an induced fit type of mechanism and that we're going to have a closing up of this rather open active site when we have substrate binding. Okay. All right. Now, what does this active site do for us? Well, the PGLC structure, as I've shown you, is rather unusual, and it actually sort of alleviates a very important problem, which is bringing together, remember, that membrane embedded substrate and a substrate from solution. And so it's putting the active site right here at the membrane interface. However, enzymes like carnitine palmitoyl transferase and other glycosyl transferases actually have their active sites far above the membrane interface. So in those cases, the enzymes had to construct a long hydrophobic tunnel to bring the membrane-embedded substrate up into the active site. But for our enzyme, that's not the case. It's right at the membrane interface. You know, it's like when you go to really one of those expensive resorts and they have the bar in the swimming pool so that you can go up and get your drink without leaving the swimming pool, right? And so it's just like that. We don't have to pull it out. Well, I wondered how common this was, right? And so we gathered together all the monotopic membrane proteins from the protein data bank, and they're shown here using, again, the PPM server to dock them. And what we found was something really interesting. That is, except in the case of our enzyme, PGLC, and this AOX enzyme, every single one of those membrane-embedded enzymes has an active site that's above the membrane interface. But they also have a soluble homologue. That is, there's a cousin of theirs that's in solution, okay? Now, why would that be? Well, nature, you know, evolving chemistry is very hard. You have to evolve a transition state, you know, a way to stabilize the transition state. You know, that is hard, something that's hard to do. Now, evolving something that can just bind to a membrane, that's a lot easier. So what we did is we overlaid the soluble homologues with their membrane-soluble cousins. And what we found was in the case of the soluble homologues, they have these extra alpha helices, which the PPM server predicts would be membrane embedded. So to make a new enzyme, don't do new chemistry. Take the chemistry that's already existing, put some helices on it, and bring it down to the membrane. You wind up with your active site above the membrane interface, but you wind up with the chemistry that you need. So this is a sort of theory of evolution. Now, what would be happening in our case is maybe our PGLC evolved more in concert with the membrane instead of starting with a soluble cousin. And so there is no soluble homologue of PGLC. All right. So that's our theory. I'm sticking to that, okay? We can discuss later. And I also wanted to get to the answer to the question that I posed before. Why would nature use two different folds to do the same reaction? Okay, so we've been looking at this fold for PGLC, but remember the polytopic enzyme does the same reaction. It does the same phosphoglycosyl transferase reaction. And it also has an active site at the membrane interface. So why have two different folds? Well, undecaproenol phosphate, the substrate, is common between these two. And these pathways occur within the same bacterial species. And so it's a rather rare substrate. It's actually only 0.1% of the lipidic fraction of membranes. So to partition that rare substrate between the different pathways, nature would have to rely on Kcat over Km or Kcat, depending on the concentration, right? So these enzymes are fighting in the two pathways, and the partitioning would just be doomed to be controlled by Kcat and Km. But if we have two different scaffolds, we might be able to have differential regulation of those two enzymes. And indeed, in our enzyme, in our Campylobacter pathways, we found evidence for both phosphorylation and regulators such as KY that are possible regulators of our pathway. We're still looking into all of this, but we think that this type of regulation could be a reason why you would need these two different folds for the same reaction. Okay. So going back to our three subfamilies, we can kind of start to look at now, can I actually discover specificity determinants for these PGTs? So when we first made a sequence similarity network, which the sequence similarity network will cluster together those enzymes that have higher sequence identity, and those that have less sequence identity will be in a separate cluster. And so, and I've seen many of these today, so you may be already familiar with those. And when we did this for our entire superfamily, all 63,000 members, really it was dominated by this large subfamily. You can see everything that's colored green here are the larges. But we were focusing in on the smalls because we wanted to start with the simplest thing, right? And so that was kind of a problem. So we needed a way to just focus in on these smalls. The smalls are down here and they look like singletons. This entire network is being taken over by the larges. So we were able to then do a size cutoff and make instead a small sequence similarity network. So this should now be only the small PGTs. There's about 32,000 sequences. And we can now try to assign these clusters to sugars. So remember, I told you for PGLC, we have NN-DINAC back, okay? And so we want to be able to assign the rest. So what we did is we made a small sugar library that's shown here, and we wanted to go ahead and test target members of the larger clusters here to see what their specificity would be, then be able to go back to the sequence and find the specificity determinants. Okay, now my lab is very, very hardworking, but they did not want to, for some reason, actually purify 30,000 enzymes. Don't even ask me about it. So what we did instead is we said, well, what if we get rid of the purification step? So what we did instead is we overexpressed the enzyme of interest in E. coli. We got the cell envelope fraction and added our sugar, our UDP sugar and our undecapropanol phosphate to the cell envelope fraction and read out using a UMP glow luminescent assay directly from that. And I guess I wouldn't be telling you about it if it didn't work. It did, okay? And what you can see is most of these enzymes actually have a fairly narrow substrate range. Some of them have a broader substrate range, but in general, they're fairly specific, as we expected they would be for this really important primary initiating step, right? And so what I was able to do then was make a phylogenetic tree and decorate the tree with the particular sugar for each of these clusters. And indeed, things tend to cluster together that are more similar and separate well into their branches. So this really, this really worked nicely to be able to do a more simplified version of a substrate specificity screen. And, were we able to get specificity determinants? We're still working on all of this, but the short answer is yes. If we take our PGLC and we take, these are AlphaFold models of other enzymes from other clusters, okay? Because remember, each of these is from a different bacteria, okay, from a different bacterial pathway. And what we can see is if I color in green here, the residues that seem to be common just within that one isofunctional cluster, we can start to make sense of this. So this ongoing work will hopefully now be able to assign all of our PGTs, well, almost all anyway, in our small family. Okay, so that takes care of our enzyme, our PGLC. Now, what do we do about one of our other questions, which is, we've got these three enzymes that can do this glycosyl transfer, and then a processive transfer of three glycosyl residues. So can we find determinants for those glycosyl transferases? They have a very different fold. They all share the GTB fold. So we solved the structure, or well, when I say we, Nino Vuksanovic in my lab solved the structure of PGLA, the first enzyme in the pathway. And I'll just show you this quick egregious movie of this. So what you can see is there's two separate Rossmann domains in the GTB fold. And here's our substrate bound into the active site. Now remember, this is the donor sugar substrate. So that catalytic site is located between the two domains. This enzyme barely dips its toes into the membrane, okay? So it's using the ppm server, you can see that we're actually very far from the membrane surface. Now our acceptor UNP and NDINAC back has to float up to this and do an SNI reaction with the substrate, you know, with the donor substrate, right? And so that was a bit of a problem right from the beginning. Okay, so we want to know then, can we explain how these enzymes, PGLA, J, and H, all take these increasingly longer glycans? All right, now let's just take a look at their structure. So here's PGLA that I just showed you. The structure of PGLH was already solved by Ramirez et al. And PGLJ I'm showing you here is an alpha fold model. Now, if you look at those again, each one is taking a longer acceptor sugar, but hey, they look really, really similar to me. Okay, let's do an overlay and zoom in on the active sites. Here are our donor sugars. This is A and the model for J overlaid. And here's A and the model for H. Again, the RMSD is quite low. These are very, very similar folds. The active sites have not moved. If I strip away the spaghetti so it's a little easier for you to see, what you can see here is that we have a very good overlay of our donor sugar. For J, again, it's just a model, but we have the specificity determinants in the same place. So this is great for us finding the specificity determinants between the glycosyl transferases among jejuni and concisus, which do differ slightly, okay? But it's not great for us understanding how these can take these ever-growing glycan chains. All right, so in a series of 3 papers, which don't worry, I'm not going to go through them all. Okay, so what we did is we found the specificity determinants for the donor sugar. As I showed you, the active sites overlay very nicely. And so, for instance, PGLJ takes either GalNAc or a GalNAc-A acid sugar. And we could find the determinants to make that switch between the two. Same thing with PGLH, we take GalNAc versus GlucNAc. But here's our acceptor sugar drawn here for PGLA, J, and then H. Right? You see that it's getting longer and longer. Here's our little schematic, our donor domain and our acceptor domain. Okay, so to answer this question, we went back to molecular dynamics. And the Straub lab was kind enough to do the same type of molecular dynamics they did for us earlier in the membrane and do a more realistic model of our protein than what we could get in just the ppm server. And so what you can see here is our undecaprenol diphosphate DINAC-back acceptor. Here's our PGLA, so the first glycosyl transferase in the pathway. And if you look at the insertion depth and the residues, what you can see is that actually, yes, the portion that's here near the acceptor domain is embedded in the membrane, but the donor domain also gets very, very close to the membrane. So it's sort of bowed down to the membrane like this so that it can reach the acceptor sugar. And you know, this reminded me of when I went to the Science Museum, and my favorite dinosaur, the T-Rex, had always been in this upright position. You know, this is how the ppm server had it, but actually the modern T-Rex is much more parallel to the ground, okay? And so it reminds me very much of that. So our donor substrate can be much closer to the membrane and to the acceptor site. Now, do I have any experimental evidence for this? Luckily, I do. So let me see if I can convince you of this. So what our collaborators in the Imperiali lab were able to use unnatural amino acid mutagenesis to put one label at a time in a protein, and then to use this tetrazine labeling reagent in a Diels-Alder-like reaction. That labeling reagent is membrane soluble. So what should happen is that anything that's in the membrane or close to the membrane will be more labeled, but anything that's farther from the membrane will be less labeled. And that's sort of shown here in this heat map. So as we felt pretty good about the using PGLC as a kind of a positive control, and you can see that those in red here are very much either very close to the membrane or membrane embedded. Those that were not labeled well are very much in the soluble portion of the enzyme. Let's do that for PGLA. What you can see is that indeed we have the membrane embedded portion being very, very labeled, and things that are very far away are less labeled. But what about this lysine here in the donor domain? It's intermediately labeled. And so it must come close to the membrane sometime in order to have that intermediate labeling. So that's my evidence for this sort of idea. So the idea is that we must get beyond the active site to really understand how some of these enzymes work. We have to mind the distance between the donor and where the acceptor will be down here at the membrane. And that's true not just for our pathway, but for other monotopic membrane proteins that are involved in these kinds of glycan pathways. And I'm just showing you here the sugar with distances in angstroms for each additional glycan that must be added. So our PGLH has to allow 3 different additions that are at about 5 angstroms from one another. So you're going to ask me, well, wait a minute, you said the structures were very much alike. How can the PGLH allow that? So in order to allow that, PGLH must have some additional flexibility or the ability to change its orientation with respect to the membrane in order to change that distance toward the acceptor. Okay, so I want to just leave you then with some perspectives of our work. Although complex, I hope that I've convinced you that oligosaccharidyl pathways and products can indeed be unraveled, and that we can determine signatures for specificity. And if we can accomplish this, we should be able to make better chemoenzymatic tools. We should be able to make glycan binding proteins better, and hopefully inhibitor discovery as well. So I'd like to thank you then for your kind attention, and I'm happy to take any questions.
It was a wonderful talk and perfectly timed for Biochem 507 students this year. So are there any questions? I guess maybe I can start us off then. So I guess I had 2 questions. One, is there any evidence that the proteins along the pathway interact with one another?
Yes, there is. So yeah, so one thing that's really interesting is if you take either nanodiscs or SMALPs and you have one of the proteins in it, it will pull down the next protein in the pathway. So PGLC can pull down PGLA, okay? But also, when we put all the enzymes together, and we do this all the time, don't forget that when I'm studying PGLH, my poor students, honestly, they have to purify PGLC, A, and J to make the substrate for H. When we do that, when we have all, so when we do that, you get the products and you don't get the intermediates. So it's as though the intermediates are not really released into solution, but they're somehow being better passed between the enzymes. We're starting on some studies, using mass photometry to get the actual complex and visualize it. And we do have some preliminary evidence that we are getting larger complexes, via mass photometry. So hopefully the next time you invite me to talk, I'll have more information about that. But we definitely have evidence for that. It's more like a metabolon.
So ironically, my poster at that enzyme mechanism meeting a long time ago was on substrate channeling.
Doesn't he sound just a little bitter about this? I'm not sure. Yeah.
I found it quite remarkable that your operon had so many enzymes that were in the same order transcriptionally and then in terms of their protein activity.
Yes.
I was wondering about the assembly of the proteins into the membrane and if there's any direction there, and whether or not if you take out, if you change the order, does that change the assembly of the proteins?
Oh, wow. Okay. I can tell you a little bit about so definitely in terms of the sequence, the PGLC, the first enzyme in the pathway, it does follow the positive in rule, and it has the kind of sequence you would expect where it's being made on the ribosome. It's going to have positive charges at the beginning of the sequence that will then go down into the membrane. And then there are positive charges on the other end where you almost would imagine it would stop then going in, right? And make interactions with the phospholipid head groups. I don't, there definitely been studies to look at, when you take the entire operon out, you get the products, even if if you move those pieces over. I don't think anyone's ever tried. That's a really good idea, is the scrambling experiment. I do know this. When I talk about the distance from the membrane and going beyond the active site, I'm serious about that. So if you take the shorter substrate of PGLA, even though GalNAc is still the apical non-reducing end sugar, okay? And you give that to PGLJ or H, they will not take it, even though it's the correct sugar, but just shorter. It won't take it. So I think that if I scrambled them, it's going to be a no-go now, but it might still make the metabolon. Right. Yeah. I love that. No, I love that, because then it would look at what the flux is, even if they were just reversed in order, and whether the order has to do with the actual assembly of the metabolon. I love that idea. I should learn to do more molecular biology. I will. I will.
Thank you for the very cool talk. I was interested in this idea that you mentioned, that you have these two isofunctional folds that give you access to maybe differential regulation. How would that work?
Yeah, what I think is that, for instance, if you have, so we know, like, for instance, encapsular polysaccharide biosynthesis, there's specific kinases in that operon that actually will phosphorylate the PGLC ortholog in that pathway. Okay, they're very specific. They do not phosphorylate the monotopic, the polytopic enzyme. So they're fold specific and they're sequence specific. So like any good kinase, right? They're looking for their correct target. And so if that pathway is altered in its specificity, such that, let's say, without phosphorylation, it's shut down. Now that undecaprenol phosphate substrate is now more available for the polytopic enzyme. So it would just affect the partitioning between the pathways. Whereas if you don't have any regulation, it's only the balance of Kcat over Km that's going to allow any partitioning, right? It's going to direct that. Does that make sense? Yeah.
You said there'd be a quiz on the PRP domain that was conserved.
Yes.
Why are the prolines there?
Okay. All right. So as I said, I don't yet have a structure with the sugars, the nucleotide sugar in it, but when we have done some really nice docking studies, okay, what you can see is the PRP motif stretches the arginine. It's as though the two prolines on either side are holding it into a rigid position, and the arginine reaches the pyrophosphate of the UDP sugar. So, and that's such an important part to stabilize for the two nucleophilic attacks that happen, right? That I think that PRP motif is just, it's completely invariant, and it makes sense that it's got to be part of the common chemistry of the.
So do you think that there's cis-trans isomerization happening,
Oh,
or just that that
okay.
the trans
I
conformation
thought
is-
about that,
is holding
but
it
I
there?
couldn't convince myself that an enzyme could reach into that active site to do that there, but maybe like earlier in folding, but definitely there's no evidence for that. But I love that idea, and that does happen in like polymerases and things like the RNA polymerases and things like that, but I don't have any evidence for it in our pathway.
Any time for one last question?
Yeah, really cool talk. I'm just curious about the processivity of PGLH.
Yes.
And if you think so with the sort of theory about the distance defining
Yeah.
some of the substrate specificity, if that's also involved in the control of how many units get added
Mhm.
by that. And if there are maybe other homologs that add different amounts.
Yeah, absolutely. So, okay, two things. One is it doesn't add a fourth, it only adds three. Like we have, no matter how much substrate you give it, it doesn't just keep going. So we know there's something about the length again, right? So that's one thing. Second of all, in Campylobacter concisus, what happened is because PGLJ takes I showed you that acid sugar. Now PGLH has to put on the next sugar after that. And so it actually made two copies of PGLH. One does that first sugar after PGLJ, and the other one does the next after that. That seems to have broken the processivity, and you actually use another enzyme after that to add the third sugar. And so somehow in evolution, once you're not adding the same sugar over and over again, it had to make a new copy, and then it was doomed to go down a different evolutionary path. Crazy. I was gonna put some more slides in about that, but I didn't want to torture you guys, but it's really cool, and it's in those papers that I pointed out.
Great. Well, let's thanks Karen for the wonderful seminar.